Okay, Assalamualaikum warahmatullahi taala wabarakatuh. So today, we are going to study a new topic, okay, which is about movement of substance across plasma membrane. Okay, so let me set up first. Okay, guys, so let's move on. Okay, hold on. Okay. So the first one, we are going to study the structure of plasma membrane. Okay, so before you want to study the structure of plasma membrane, you should know what is the function of plasma membrane. Okay, during your Form 1 and Form 2, I'm sure you guys use the term cell membrane, right? Okay, from now and on, you should use the term, okay, plasma membrane instead of cell membrane. Okay, plasma membrane and cell membrane are the same thing. Okay, so basically this is how plasma membrane looks like. Okay, so if you see here, plasma membrane, okay, can be made up by two things. Okay, the first one is phospholipid bilayer here. Okay, if you see the blue color, and then it has tail. Okay, at the top and also at the bottom. Okay, both this thing, okay, top and bottom is called as a phospholipid bilayer. Okay, and then the second one is protein. Okay, so I repeat again. Plasma membrane is made up by two things. The first one is phospholipid bilayer. The next one is protein. Okay, guys. So if you've noticed protein, there are various type of protein. First one, we have carrier protein. Okay, and then we have channel protein. We have glycoprotein. Okay, so protein we have how many types? 1, 2, and 3. So how can you distinguish between them? Okay, so for channel protein, if you notice, there is a way. Ni ada jalan kat sini ni. In the middle. Okay, but for carrier protein, okay, I think this structure is not really suitable for us to study, but the shape of carrier protein, it looks like this. Let's see. This. Okay. This is carrier protein that we discussed just now, where I said there is a way. Dia ada lubang kat sini kan. Where this will make the substance can easily get in and easily get out from the cell. Okay, but for carrier protein, if you notice, this is how it looks like. It has a very tiny hole, okay, in the middle. But here, you see, dia ada jalan straight forward. But here, for carrier protein, there is a hole inside where it need to change its shape. Okay, so we can say that carrier protein, it will change its shape in order to allow the substance to get in or get out. Saya tengok kat sini ni. Okay, here the substance would like to get inside carrier protein. So carrier protein dia akan ubah the structure. And then this thing, it will bind to the hole. Here we call it as a active site ataupun binding site. We do not call it as a hole. Okay, we call it as a binding site. Okay, tempat dia melekat tu. So it need to change another shape to allow the substance to get into the cell. Okay, can you understand? Okay, guys. So we done for carrier protein. Okay, carrier channel protein. The last one is glycoprotein. So if you see, this is protein. Okay, and this is carbohydrate. Okay, so Glycoprotein is a combination between protein and carbohydrate. Okay, can you understand? Okay, so if just like this, okay, it doesn't match with anything, so we call it as a glycolipid. Yang ni dia gabung dengan protein kan, so we call it as a glycoprotein. And then this is glycoprolipid sebab apa? Ni kan phospholipid bilayer. So this is carbohydrate. So glyco stand for carbohydrate, lipid stand for the lipid. Same goes here. Glyco stand for carbohydrate here. Protein is this one, protein. Okay guys, so the next one is about phospholipid bilayer. Just now we know that plasma membrane is made up by two, phospholipid bilayer and protein. Protein we have discussed just now. Now we want to focus on phospholipid bilayer. Okay, if I make it bigger, okay, so this is how one single phospholipid looks like. So it's made up by head, kepala, and tail. This is tail and this is head. Okay, so head and tail ni they have characteristic. Okay, first one, head ni dia adalah hydrophilic. Okay, head ni dia hydrophilic. What does hydrophilic means? Hydrophilic means it attract water. Okay, I repeat again. Head ni it is hydrophilic. Hydrophilic means it can attract water. Dia akan attract water. Okay, but for tail, they are hydrophobic. Okay, so tail are hydrophobic. Okay, try to guess. Kalau yang ni hydrophilic, attract to the water. Kalau hydrophobic, okay, so tail is hydrophobic. Hydrophobic means that it does not attract water. It does not attract water. Instead, it attract fat. I repeat again. So tail ni dia adalah hydrophobic. So hydrophobic it will attract fat, not water. Okay. And then the second one you see here, polar and non-polar. Okay, head is polar. Why? Because it is placed at facing in outside the cell and facing inside the cell. Okay, ni adalah plasma membrane, right? So this is the inner cell. This is the outer cell. Ni bahagian dalam sel, ni bahagian luar sel. So if you notice, the head is facing outside the cell and inside the cell. Maksudnya dia ada arah tuju, ke luar dan ke dalam. Tapi kalau non-polar, why? Because it is put in the middle, dekat tengah-tengah ni. That's why tail we call it as a non-polar, but head is polar. Okay, guys. Oh, sorry. Okay, guys. So just now, you have to know that what is the function of plasma membrane. Plasma membrane fungsi dia apa? Fungsi dia adalah to allow, some substance move into and out of the cell. Okay, I repeat again. The function of plasma membrane is to allow some. Okay, awak kena emphasize the word some. Ataupun if you don't want to use the word some, okay, you can use the word certain. Certain, maksudnya sesetengah saja. I repeat again. The function of plasma membrane, okay, is to allow some substance to move in and move out from the cell. Okay, this substance, okay, for example, oksigen, carbon dioxide, water, apa lagi? Glucose, amino acid, vitamin and ion and so on. Okay, that's why it only emphasize the word some. Okay, kalau cakap all, we would say that bacteria also can get into the plasma membrane. Tak boleh lah macam tu sebab tu dia cakap some. Maksudnya substance yang berguna saja will get in and get out. Okay, can you understand? Tengok ni. Okay, this is the one that I have discussed with you just now. Okay. Ni, there are various types of protein embedded partially or fully within the plasma membrane. Kalau ini embedded fully lah kan. Kalau embedded partially, contohnya yang ni glycoprotein. Dia tak melekat sepenuhnya dekat sini. Dia akan melekat on the surface sahaja. Okay. So just now we have studied the function of channel protein and carrier protein. So channel and carrier protein ni fungsinya is to allow substance ataupun molecule to get into the cell or get out from the cell. Okay, how about glycoprotein? Ni glycoprotein ni. Apa fungsi dia? It act as a receptor. Okay, act as a receptor to hormone such as insulin, okay, stabilize membrane by forming hydrogen bond with the water and act as an antigen, okay, for cell and identification. Okay, guys, so this is the function of glycoprotein. Okay. Okay, there is also cholesterol, okay, found between phospholipid bilayer. So here you can see this is cholesterol. Okay, so what is the function of cholesterol? Tengok kat sini. Okay, so cholesterol will make the phospholipid bilayer stronger and more flexible and less permeable to a water-soluble substance such as ion. Okay, so kolesterol dekat sini. Okay, sorry. This cholesterol, okay, the function is to make the phospholipid become stronger. Okay, apa lagi? Stronger dan juga more flexible and less permeable to a water soluble substance. Okay, water soluble substance ni maksudnya adalah contohnya adalah ion, glucose and so on. Why? Because ion, glucose and so on, they will use protein instead. Rather than phospholipid bilayer, it will use protein. So contohlah kalau dia not water soluble, not kalau dia more permeable to a water, maksudnya ion pun boleh lalu kat sini lah. Okay. So yang boleh lalu kat ion, glucose, amino acid and so on, they will use protein. Okay, not phospholipid bilayer. Okay, guys. I think that's all for this page. Okay, let's go to the next one. Okay, permeability of plasma membrane. Okay, what does permeability means? Okay, let's go. Okay, before we want to study this subtopic, you should know what does permeability means. Okay, this permeability. Okay, I will tell you right now. Okay, permeability. Okay, I don't want to use the words here. Okay, I want to give my own definition, which is much simpler. Okay, permeability. Okay, is an ability to allow all all substance to get in or get out from the cell. Okay, I repeat again. Permeability is an ability to allow all, kena emphasize perkataan all, semua. All substance, okay, all substance, substance tu tak kisahlah oksigen ke, carbon dioxide ke, water ke, apa ke, dah all substance to get in or get out from the cell. Okay, but right now you have to know that plasma membrane, okay, it is not fully permeable. Instead, it is selective permeable ataupun it is partially permeable ataupun kita boleh cakap it is semi permeable. So plasma membrane ni, okay, dia punya karakteristik yang pertama, you can use the word selective permeable. Lagi you can use the word partially permeable. Lagi awak boleh guna perkataan semi permeable. Apa maksud partially, selectively dengan semi, cikgu? Maksudnya adalah it will only allow some ataupun certain substance. Maksudnya dia tak menyebabkan semua benda boleh masuk. Some ataupun certain substance can pass through it. Itu maksud dia. Okay, can you understand? So if we want to talk about permeability of plasma membrane, we would say plasma membrane is selective permeable, or plasma membrane is semi-permeable, or we can say plasma membrane is partially permeable. Why? Because it only allows some substance to move in and move out through them. Okay, boleh faham? Okay guys, so let's continue. Okay guys, so the next one, we want to study about the substance that can move in and move out from plasma membrane. Macam tadi cikgu beritahu as a general, substance tu could be oxygen, amino acid, water, glucose and so on. Okay, now we are going to study in detail. Okay, so characteristic of movement substance across plasma membrane. So the substance can be divided into two. Either they are lipid soluble substance or lipids insoluble substance. Okay. Okay, this one is lipid soluble substance. This one is lipid insoluble substance. Okay, other than lipid insoluble substance, you can replace with the term water soluble substance. Kalau ni lipid soluble substance, yang ni water soluble substance. But here they use the term lipid insoluble. Yang ni soluble. Ini larut, ini tidak larut. Okay, but other term you can use water soluble substance. Okay, but I don't know why this book use this term, but it's alright. They are the same. Okay. Okay, kita kisah satu. Kalau lipid soluble substance ni, okay, these are the example. Okay, fatty acid, glycerol, fat soluble vitamin, which are A, D, E, and K. Okay, vitamin adik. Okay, vitamin A, vitamin D, vitamin E, and vitamin K. And the last one is steroid component. Okay, bear in mind, all these lipid soluble substance, they will use phospholipid bilayer to get in and get out from the cell. You see here ni, lipid soluble molecule. Ni awak tengok yang warna kuning ni. Okay, lipid soluble molecule refer to all these things. Fatty acid, glycerol. For example, this is fatty acid. So this fatty acid, they want to get inside the cell. Ni extracellular environment ni kat luar sel. Ini cytoplasm. Maksudnya ini adalah dalam sel. So glycerol tu can easily get into the cell using phospholipid bilayer. Sebab ni phospholipid bilayer. Yang ni protein, ni pun protein. Ni carrier protein, ni channel protein. Boleh faham? Okay, sama juga dengan glycerol, vitamin and so on. So they can just easily diffuse inside the cell using phospholipid bilayer. And this process is called as a simple diffusion. Okay, simple diffusion. Nama proses yang mana lipid molecule such as fatty acid, glycerol, vitamin semua ni get inside the cell is called simple diffusion. Okay, tak apa. Paling kita belajar lagi. Okay, the next one. Okay, kita pergi kepada sini. Lipid insoluble substance. The other name is water soluble substance. Water soluble substance. Okay, dia terbagi pada dua, which are small molecule ataupun ion or large molecule. Okay, kita pergi satu-satu. So, awak tengok sini. Kalau contoh large molecule, for example, glucose and amino acid. So, they will use protein instead of phospholipid bilayer. Awak tengok kat sini. So kalau large molecule, they will use protein. Specifically apa? Carrier protein. Large molecule contohnya apa? Glucose and amino acid. So ni contohnya adalah amino acid ataupun glucose. So they will use carrier protein rather than channel protein. Mungkin sebab lubang dia kecil kat sini. Boleh faham? So the next one is small molecule dan juga ion. Okay, so they are polar molecule. Example dia adalah water. So water, they will use phospholipid bilayer. Sama macam ni. Sama macam lipid soluble substance. Okay, water ni special sikit, dia akan guna phospholipid bilayer. Okay, and the process where the water get in or get out from the cell using phospholipid bilayer is called osmosis. Okay, nama proses yang mana air bergerak keluar masuk menggunakan phospholipid bilayer is called osmosis. The next one is non-polar molecule, which are oxygen and carbon dioxide. Okay, so ni awak tengok ni, O2 dengan CO2. Oxygen akan masuk, carbon dioxide akan keluar. They also will use phospholipid bilayer. Boleh faham? So they also will use phospholipid bilayer, and this process is known as a simple diffusion. Sama proses dengan yang ni. Yang ni orang panggil simple diffusion. Yang ni osmosis. Yang ni simple diffusion. And the last one is ion. Ion. Okay. K plus and A plus. Okay, special for ion ni, they will never use the phospholipid bilayer. Instead, they will use channel protein. Sebab dia kecil, lubang dia kecil kat sini kan. So, they will just get inside or get outside. Ni awak tengok ni, small molecule ataupun ion, dia akan guna channel protein. Okay guys, can you understand? If you don't understand, please rewatch this video and make some note until you understand. Okay, let's go. Okay guys, so this is the experiment that basically we will do in the lab, but unfortunately, since we are doing online class, so it's hard for us. Okay, it's alright. So I will explain to you how to do what experiment is all about, what you should know. Okay. Okay, so here, okay, kita tengok dulu apa dia punya problem statement. Does the size of dissolved particle affect the movement of substance across selectively permeable? Okay, so sekarang ni mungkin kita ada dua size yang besar dan size yang kecil. So will this affect the movement of substance across selective permeable membrane? Okay, so we have this thing. Okay, we have beaker. Inside beaker, we have distilled water. And we have whisking tube. Whisking tube ni adalah tube. Tube yang mana it can replace plasma membrane. Ataupun we can say that whisking tube ni, it has the same characteristic like plasma membrane, which is semi-permeable. Whisking tube ni. Selalunya kita akan guna whisking tube lah to represent plasma membrane. Sebab apa? They share the same characteristic, which is semi-permeable. In the exam, maybe they can ask you, why we use whisking tube? So your answer, you can say, because whisking tube, okay, has the same function, has the same characteristic like plasma membrane, which is semi-permeable. Okay, inside whisking tube, okay, ada dua benda yang diletak, which are glucose and starch. Okay, so I want to ask you, between these two, which one is bigger, which one is smaller? Okay, cikgu tahu semua tahu ni kan. Tanya adik awak kat Tadika pun tahu kan. Okay, so glucose is small, starch is big, right? So cikgu nak tanya awak, contohlah kat sini kita ada water, we have whisking tube. Inside whisking tube, we have glucose and starch. Okay, between glucose and starch, who will get out? And who will remain in whisking tube? Okay, the answer is glucose will get out, but starch will remain in the whisking tube. Why? Because we know that whisking tube ni only allows some substance to get out from the cell, from the here lah. So, okay, starch will remain. Why? Because starch is big in size. Okay, and then glucose is small in size. So that's why glucose can escape from the whisking tube, while the starch will remain. Okay, so awak dah nampak kan. So at the end of the experiment, we would say glucose ni get out. Dia maksudnya glucose ni dia akan bercampur dengan distilled water, but this starch ni it will remain in the whisking tube. Okay, so awak tengok ni the result. Okay, so this is the initial. Okay, this is the initial observation. Yang mana kita tahu content dalam whisking tube ni kita ada glucose and starch. Content dekat beaker kita ada distilled water. Okay, so after the experiment, okay, what will you see? Okay, inside the beaker kita boleh cakap ada distilled water dan juga glucose solution. Tapi dalam whisking tube hanya ada starch suspension. So if you want to study the presence of starch and glucose, we can do this thing. This test. Iodine test ni untuk test kehadiran starch. Okay, Benedict test ni untuk test the presence of the glucose. Okay, so if there is a starch, it will change the color from yellowish brown to the dark blue. If there is no starch, it will remain yellowish brown. But for Benedict test, if there is a glucose, bridge red precipitate will form. If there is no glucose, no red bridge precipitate will be formed. Itu saja. Okay guys, so that's all. Okay, let's go to the next one. Okay guys, so for this experiment, I want you to read by yourself. Okay, this one apa dia punya problem statement? How do water molecule permeate across permeable membrane? Okay, here, okay, I want you try to read on your own and try to guess what the experiment want to tell you. Okay, maybe you can pause the video and you read on your own. Okay, if you don't understand, you can PM me. Okay, same goes here. Okay guys, so the next one, we are going to study about the name of the process that the substance get in and get out. Macam tadi cikgu cerita, simple diffusion, osmosis. Now we have very new term, okay? Facilitated diffusion, active transport. Okay, so now we are going to study in detail. Okay, so please pay attention. Okay. Okay guys, so let's continue. Okay. Okay, now we are going to study about the process, the name of the process, okay, where the substance get in and get out. Okay, so transport, okay, awak tengok transport ni is divided into two. Okay, the first one is passive transport. Okay, the next one is active transport. Okay, so transport ni ada dua, transport positif and transport aktif. So passive transport ni ada tiga jenis. Okay, wait. Okay, so there are three examples for passive transport. The first one, simple diffusion, and then osmosis, and the last one, facilitated diffusion. Okay, so we go one by one, okay? So what does simple diffusion mean? Okay, before we want to study, I want to say the substance that will use simple diffusion, okay, first one is, ni awak tengok ni, these are the example of substance that will use simple diffusion. The first one, oxygen, carbon dioxide, fatty acid and glycerol. Remember, gas, fatty acid and glycerol. This is gas, this is lipid soluble molecule. So they will use simple diffusion. Why? Because they can easily get inside the cell across phospholipid by bilayer. Okay, tengok. Simple diffusion is the movement of molecule or ion. Okay, I don't know why they put ion here, but I think this one should be replaced by gas. Okay, molecule, lipid molecule and gas sepatutnya. Okay, because here we cannot see any ion, right? So I think this is typo. Okay, I repeat again. Simple diffusion is the movement of fat molecule or gas from area of higher concentration to area of lower concentration. Okay, for example, okay, kat sini kita tengok ada banyak carbon dioxide. Betul tak? Ada berapa carbon dioxide ni? 1, 2, 3, 4. So inside the cell, how many? 1, 2. Okay, 1, 2. So we would say, okay, kita tak nak guna carbon dioxide lah sebab macam pelik je example dia. Okay. I repeat again. Sorry. Okay, simple diffusion is the movement of fat molecule ataupun gas from a higher concentration to a lower concentration. For example, okay, kita guna fatty acid lah sebagai contoh. Okay, fatty acid kita tengok ada berapa kat luar ni. Ni kat luar, ni kat dalam kan. Okay, fatty acid ada berapa? 1, 2, 3, 4. So we can say that we have higher concentration of fatty acid here. Okay, but kat dalam ni ada berapa? 1 dan 2. We have lower concentration of fatty acid. So that's why fatty acid can move from a higher concentration to a lower concentration. Same goes to the oxygen. Oxygen can travel from higher concentration to a lower concentration across phospholipid by bilayer. Okay, and then we can say that the moving molecule are said to move down concentration gradient until dynamic equilibrium is achieved. Down concentration gradient maksudnya, awak tengok kat sini concentration dia tinggi, kat sini concentration dia rendah. So daripada high ke low, down lah. Down concentration. Okay, and this will happen until dynamic equilibrium is achieved. Dynamic equilibrium tu maksudnya, kalau kat luar ni, awak tengok ada 4 contohnya. Kat sini ada 2. So, maksudnya, satu je lah yang kena datang kan. Sebab apa? Bila yang ni, kita ada 4. Okay, 1, 2, 3, 4. 1, 2. So, kalau satu datang sini, awak tak rasa ke sini akan jadi 3, sini pun akan jadi 3. So, once they have achieve dynamic equilibrium, then the simple diffusion will stop. Maksudnya sini ada 3, sini ada 3. So dynamic equilibrium tu. Okay, can you understand? Okay, the next one is osmosis. Tengok osmosis. Bear in mind, osmosis will only involve the movement of water. Okay, the movement of water. Sama juga macam simple diffusion. Okay, where osmosis refer to the movement of water from high concentration of water to a lower concentration of water. Okay, water potential dengan water concentration are the same. Okay, but here to avoid you from being confused, you can use water concentration lah ke perkataan air tu. Okay, so I repeat again, osmosis is the movement of water across plasma membrane from higher water concentration to a lower water concentration area. Okay, so for example here, okay, awak tengok kat sini. Ini higher water potential, yang ni lower water potential. Sebab apa awak tengok kat sini? Ada sucrose molecule. Okay, sucrose molecule, how many we have? 1, 2, 3, 4, 5, 6. Ini ada berapa? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, 13. So, I would say that here it has higher water concentration. Sebab apa? Sebab dia ada kurang sucrose. Yang ni ada low water concentration. Sebab apa? Dia ada banyak sucrose molecule. Dia terbaliklah. Kalau yang sini pekat dengan sucrose, yang ni pekat dengan air. Boleh faham? So right now we have study osmosis ni dia akan berlaku daripada higher water concentration to a lower water concentration. That's why more water from this area will move to this area. Sebab tu awak tengok ni error ni menunjukkan there is a movement of water from here to here. Okay, sucrose tak bergerak water saja yang bergerak. Sebab apa? We are talking about osmosis. Okay, so water will move from this area to this area by osmosis. So that's why at the end of the experiment you see the level of water this area is lower than here. Why? Because more water diffuse from this area to this area. Boleh faham? Okay guys, so let's go for facilitated diffusion. Okay, awak tengok kat sini. Okay, this is facilitated diffusion. Okay, for facilitated diffusion, they can use two. Okay, wait. It's alright. Okay, guys. So for facilitated diffusion, they can use both protein. Either they want to use channel protein ataupun carrier protein. But bear in mind, the process is different. The process is different. Awak tengok ni, kalau channel protein ni yang terus pergi ni, awak tengok. Form a channel or canal to allow small size solute or ion diffuse across plasma membrane easily. Maksudnya dia akan masuk macam tu saja. And channel have a specific internal characteristic that only allow specific ion to pass through it. Maksudnya walaupun dia adalah channel ataupun ruang yang terbuka, it will only allow certain, tak semualah, ataupun allow specific ion. Maksudnya bakteria tak boleh lalu kat sini. Lepas tu carrier protein ni, awak tengok ni carrier protein ni. It has a specific site. Tengah ni orang panggil specific site. Okay, that bind with a specific molecule. For example, glucose only bind to a specific site of glucose of carrier protein. Okay. Okay. So this is the example of facilitated diffusion using carrier protein. Dia using carrier protein. Okay. Kalau yang ni tak payah bincang lah sebab dia just diffuse in and diffuse out easily. But for carrier protein, there is a specific mechanism. Okay, kita pergi satu-satu. Okay, pertama sekali awak tengok glucose ni. Okay, the concentration of glucose outside is higher than inside. Sebab itulah it can travel inside the cell. Okay, awak tengok. Extra glucose concentration are higher than in the cytoplasm. So, apa yang terjadi? So, the glucose will bind to the specific site. Kat tengah ni kan ada lubang kan? Okay, carrier protein ni ada lubang. Nama lubang tu adalah specific site. So, glucose will bind to a specific site of the carrier protein. And then what happen? The carrier protein will change its shape to allow the glucose pass through carrier protein and enter the cell. Boleh faham? And then carrier protein return to its original shape and ready to transport another molecule. And then this will start all over again. Dia akan proses yang sama akan berlaku. Boleh faham? Okay guys, so ni awak tengok kat sini yang tadi cikgu tak bincang lagi ni yang information facilitated diffusion. Okay, so facilitated diffusion, okay, contoh substance yang akan guna dia adalah ion, okay, amino acid, glucose. Okay, so they will use either carrier or channel protein. Okay, facilitated diffusion does not require energy because the transport protein down concentration gradient. Down concentration gradient macam tadi lah. Okay, where the substance will go from higher concentration to a lower concentration until dynamic equilibrium is achieved. Okay, where the concentration of molecule is the same at both sides. Okay, sama macam tadi lah. Ingat yang ni tak menggunakan tenaga. Okay, the next one is active transport. Active transport ni dia tak ada contoh. Satu saja contoh je, iaitu active transport. Okay, apa karakteristik dia? Okay, kita pergi satu-satu. Okay, the movement of substance across plasma membrane may occur by active transport. Okay, now we have done passive transport yang ada 3 tu. Simple diffusion, osmosis dan juga facilitated diffusion. Now we go for active transport. Okay, so the characteristic of active transport, okay, ini contoh dia. Okay, ini karakteristik dia. The movement of molecule or ion substance across plasma membrane occur against concentration gradient. Okay, macam facilitated diffusion dan juga osmosis, kita tengok dia cakap down concentration gradient kan, tapi yang ni against concentration gradient. Apa maksud dia? Kalau down concentration gradient, maksudnya higher concentration dia akan pergi dekat lower concentration. Tapi ini terbalik. Maksudnya daripada yang sini dah lower, yang sini higher, yang lower pula pergi kat higher. Maksudnya there is imbalance. Imbalance dynamic equilibrium. Itu maksud dia. And then it require ATP energy. Dia memerlukan tenaga. Sebab tu dia aktif. Nanti kita cerita lagi. And then it requires specific carrier protein with a specific site to bind a certain molecule. Sama juga macam ni. Dia ada carrier protein yang mana dia ada specific site. And carrier protein poses receptor to bind with ATP energy. And carrier protein change its shape. Ni carrier protein ni change its shape. Okay, when the phosphate group attached to it, as the result, the the molecule or ion can move across the plasma membrane. So these are the characteristic of active transport. Okay, now, okay, we want to study the process. Okay, kita tengok satu-satu Okay, so the first one you see here, tengok. This is the outside the cell, this is the inside of the cell. The proof is here, ni cytoplasm. Maksudnya dalam sel ni. Okay, macam tadi kita tengok, dia cakap active transport ni, dia against concentration gradient, right? Sebab apa? It travel from lower concentration to a higher concentration. Awak tengok. Yang ni dah banyak dah, yang ni sikit. Tapi yang sikit ni nak pergi juga yang tempat banyak tu. So, it against concentration gradient. So, awak tengok kat sini So, pertama sekali, 3 sodium bind to the carrier protein. Ni, awak tengok. Ada 3, 1, 2, 3. Ni dah sikit dah. Lepas tu dia nak pergi tempat banyak pula. Lepas tu ATP energy. ATP energy ni is given by mitochondria. Sebab kita tahu dalam mitochondria tu dia akan hasilkan ATP energy. So ATP energy tu akan ATP energy ni datang daripada mitochondria. So this ATP energy will go nearby to the carrier protein. So apa ATP energy tu buat? ATP molecule will break. Dia akan pecah kepada ADP dengan P. Okay, so ATP energy ni dia akan berpecah jadi 2, ADP dengan P. So apa terjadi? Phosphate group ni dia akan bound to the carrier protein. Dia akan melekat dekat dia akan melekat dekat carrier protein. Okay, so apa yang akan jadi? Phosphate bond provide energy and change the shape of carrier protein. So once the phosphate attached to the carrier protein, this can give energy to the carrier protein to change its shape. Okay, so this will allow the sodium to be transported, get out from the cell. Boleh faham? Okay, ini contoh pertama. Ini contoh yang kedua pula. So kita tengok kat luar ni. Kat luar ni potasium dah sikit, tapi kat dalam ni dah cukup banyak dah. Tapi masih lagi, that's why we can say it is again concentration gradient. Sebab apa? Dia tak kisah pun pasal gradient yang mana yang rendah, gradient mana yang tinggi, concentration. Okay, tengok. Two potassium from outside bind to the carrier protein, specifically in that specific site. Okay, lepas tu phosphate group leave the carrier protein. Dia akan tinggalkan. So, the loss of phosphate group will store the original shape of carrier protein. So, bila phosphate group ni, dia detach from carrier protein, okay, this gives signal to the carrier protein to return its original shape. So, bila dia return to original shape, this can allow the potassium ion to get inside the cell. Sadaqallahul azim. Boleh faham? Okay guys, so okay, awak tengok ni. Ni contoh lain pula ni. There is another pump known as a protein pump. Okay, for example, proton pump are found in epithelial cell lining in a stomach cavity. Protein pump cause the acidity of the stomach content. Okay, so apa ni nak cakap apa sebenarnya ni? Kejap. Okay, ni cytoplasm. Okay, yang ni adalah dekat luar sel. Okay, so the energy from ATP energy from ATP enable hydrogen ion to be transported, okay, by carrier protein towards extracellular fluid. Okay, and this cause accumulation of hydrogen ion and acid production in a stomach cavity. And protein pump is described in the figure 3.10. So tengok kat sini. Ion also, okay, tadi kita bincang pasal sodium dan juga potassium ion. Now we are talking about hydrogen ion. So hydrogen ion pun samalah, dia akan pergi daripada sini ke luar. So against concentration gradient. Tengok. Kat sini dah lah sikit dah, sini dah banyak tapi dia pergi juga. Okay. Okay guys, so this is the comparison, okay, between passive transport and active transport. So kalau warna hijau ni, this show to you the similarity. Okay, similarity between passive and active. Tapi yang sebelah kiri dan sebelah kanan show the different. Okay, kita pergi similarity dulu. Okay, ingat similarity you should use the word both. Okay, both moving a substance across plasma membrane. Okay, betul lah. So both occur through selective permeable membrane. Okay, betul. Okay, kalau perbezaan dia, okay, active transport require energy, specifically ATP energy. Okay, tapi kalau passive transport, okay, it is not, okay, energy is not required. Boleh faham? Okay, kalau active transport, it occur against concentration gradient. Tapi kalau passive transport, it occur follow the concentration gradient ataupun it occur down concentration gradient. Sama juga. Follow concentration gradient ataupun down concentration gradient pun you can choose either one. And the last one, active transport, okay, there is a accumulation and disposal of molecule or ion. While passive transport, it will occur until dynamic equilibrium is achieved. Sebab apa accumulation or disposal of molecule? Sebab awak tengok ni. Sebab ni awak tengok ni. Okay, Na ni dia banyak pergi kat sini kan. So we can say that there are so many accumulation of Na outside the cell. Sebab apa? Mungkin kita nak buang lah Na ni sebab terlalu banyak. Okay, disposal kita nak buang. Okay guys, so I think that's all for today's class. Okay, for the next topic, we are going to study about this one. How can we apply this in our living organism? Okay, so these are the things that you are going to study on the next class. Okay, can you understand? Okay guys, so that's all for today. So if you have any question, you can PM me. So make sure You guys write down the name and class for attendance purpose. Okay. So if you have any question, you can ask me. Okay, that's all for today. So I see you on the next class. Bye bye. Assalamualaikum.